who are focused on understanding the molecular drivers of this memory-robbing disease. Their goal is to identify targets and strategies for developing treatments and preventative methods to control the disease. So these seminars are interactive. As moderator, it's great fun to include your questions in these conversations. It also helps me to identify topics that are of the greatest interest. So there is a Q&A button at the bottom of your screen. Please go there to type in your questions. And a quick disclaimer. All of the scientists in today's events are PhDs. They are not MDs, so they are not qualified nor licensed to give medical advice. So please don't bother us with those kind of questions. So this is how the event's going to work. I'm going to do brief introductions for each of the professors. There's more information about their backgrounds on the website if you're interested in that. They are going to give an overview of their work. Then when all three have had their chance at bat, we will start the conversation, and that's when we will include your questions. So first up is Dr. Julie Andersen, who trained as a neuroscientist. Julie's lab is focused on understanding the age-related underpinnings of both Alzheimer's and Parkinson's diseases. Here's what I think is cool. Some of the work in Julie's lab is focused on boosting natural protective processes that tend to go haywire as we age. And one particular project involves looking at a metabolite that is derived from fruit as a possible treatment for Alzheimer's. Julie, are you online? You want to tell us about what you're up to? Do we have Julie online? I don't. You know what? You're going to be up next. Let's move on to Tracy, then we'll come back to Julie. So Tracy is also a neuroscientist. Her team is particularly interested in how toxic proteins called tau impact synapses. Those are the specialized gaps between neurons that are critical for information to be transmitted throughout the brain. One of Tara's projects is focused on how the brain can be resilient to the memory loss caused by these age-related toxic proteins. So Tara, welcome. Why don't you get started, and we'll make sure to bring your slides up. There you go.
Thank you, Kris. It's really a pleasure to be here to tell everyone who's joining us about the research going on in my lab here at the Buck Institute. So I'm really happy to be here and happy to answer questions from you all. Next slide, please. Thank you. So Alzheimer's disease is characterized by the accumulation of two toxic proteins in particular in the brain called amyloid beta and tau. And you can see here in this illustration on the left side, on the left panel, is a picture of normal looking neurons in the brain. And on the right side, we see neurons that contain pathological neurofibrillary tangles, which are comprised of toxic tau proteins. And then we have amyloid plaques, which are comprised of the amyloid beta protein. And so my lab in particular is interested in understanding what the toxic tau protein is doing to the neurons to affect their function in Alzheimer's disease. Next slide, please. And one of the important things that we think the toxic tau is doing to the neurons in the brain is affecting their synapses. The synapses, as Kris pointed out, are these very, very small specialized structures in the brain. There are over 100 trillion synapses in your brain, and they facilitate the transmission of information from one neuron to the next. And the activity at these synapses, as shown on the right, the activity here across synapses is critical for the encoding of new memories in your brain. So We think that these synapses are impaired in Alzheimer's disease, and that leads to memory loss in the disease. Next slide, please. And so my lab in particular is interested in understanding how these synapses are dysfunctional in disease. And we think this is really important because, as you can see here, as toxic tau builds up in the brain, we see a cognitive decline in disease. And we think that one of the earliest things that happens when toxic tau builds up in the brain is synapse dysfunction. And then later on, we think those synapses become lost. And finally, we have neurodegeneration, the neurons die. So if we can figure out how synapses are dysfunctional early in the disease, we think we can slow the progress of cognitive decline as early as possible. Next slide, please. And so my lab in particular is interested in whether we can make synapses resilient to the toxicity caused by tau in Alzheimer's disease. And we think this could be possible because there are some studies on humans showing that about 8 to 12% of human subjects can have amyloid beta and tau accumulation in the brain, suggesting that they have Alzheimer's disease. However, we think these individuals could be resilient because they do not actually have memory loss that should occur with Alzheimer's disease. So we think this is a possibility that we could have in future a resilience therapy. Next slide, please. So my lab is working on a protein that's found at synapses called qibra. And we think qibra could be important because we think it may modulate the resilience and vulnerability to memory loss in Alzheimer's disease. And we think this because qibra is an important protein for memory formation. And I previously found that reduced qibra levels, as shown here in this picture of synapses, reduced qibra levels in the Alzheimer's disease synapses occurs with memory loss. And so now my lab is working to see how we can enhance the qibra protein and signaling at synapses as an approach to protect against tau-induced memory loss in Alzheimer's disease. So thank you for your attention, and I'll look forward to discussing more about Alzheimer's disease.
That's great. Thanks, Tara. So thank you. I'm seeing great questions come in, so keep them up. I look forward to having the conversation. Next, we're going to go to Pankaj Kapahi. So Pankaj is not a neuroscientist. I am betting that when he came to the Buck in 2004, he never thought that he would be studying Alzheimer's disease. Dr. Kapahi was trained as a geneticist. His team is focused on the role of energy, nutrition, and energy metabolism as it relates to aging and age-related diseases. His lab studies the impact of caloric dietary restriction. I get that wrong. It's dietary, not caloric restriction, which he says sometimes makes him the least popular guy at neighborhood barbecues. Why don't you talk to us, Pankaj, about your work with Alzheimer's?
Thank you very much, Kris. I'm really excited to be here and participate in this webinar. And thanks to Tara for that beautiful introduction. It sets me up for one of the next questions that I'm going to try to answer. And that is the question of what are the risk factors for Alzheimer's disease? And two of the major ones we're interested in are aging and sugar metabolism. Could I have the first slide, please? So I want to start by saying, Tara pointed out, showed how, how we have the trillions of synapses. One of the important things to remember is that sugar is the main substrate for energy for neurons. And yet we have a very interesting paradox that on one hand, sugar is the main source of energy for neurons, but on the other hand, sugar metabolism, when disrupted, is now being linked with Alzheimer's disease. So, and we're interested in this question, how both aging and disruption of the glucose metabolism influences Alzheimer's disease. So one of our hypotheses is that it's not sugar. For example, on one hand, more high sugar levels are toxic, just like in diabetes, and diabetes is one of the risk factors for Alzheimer's disease. If you have diabetes, the risk of developing AD and other dementias can go from 2 to 4-fold. We think it's not glucose itself that is toxic, but something we call methyl glyoxal, which is a downstream product of when sugar is utilized in the cell that is toxic. This binds to proteins, DNA, and lipid, and can cross-link. And you can see this complicated molecule, how sugar leads to this very reactive molecule, methyl glyoxal, and becomes this complicated cross-linked molecule, which then wreaks havoc on different parts of the body and is being associated with a number of age-related and diabetic pathologies, including diabetic retinopathy, nephropathy, neuropathy. But importantly, higher levels of these agents are also found in dementias like Parkinson's and Alzheimer's disease as well. So, and one of the other things I wanted to point out is you may have heard the term ages first time today, but you're all familiar with, because every time you cook, that's the reaction we are practicing. It's considered the most commonly practiced reaction by humans, because you mix sugars, amino acids and sugars, and when you heat it up, you form advanced glycation end products. And this is what leads to this very tasty flavor. And one of the interesting things our lab is sort of also asking is, does the production of ages also makes you overeat and makes this a sort of a positive feedback, sort of a positive feedback loop? So you build more ages, you end up overeating, leading to metabolic disturbances and also increased risk of dementia. Next slide. So to cut a long story short, we have come up with a concoction of commonly used, FDA approved supplements, which are, safe to use in humans, which is called Glilo. For glycation lowering. And we have tried that in a number of settings to see and found very interesting and protective effects of them. So for example, this is a mouse which has a mutation in the leptin pathway. So leptin is very important for food regulation. The mouse almost doubles its weight. We give the mice this, we treat them with Glilo, and you can see a normal mouse will gain weight, but if you put it on Glilo, it actually reduces weight and it levels off to a different set point. And we have found that not only does it reduce weight, but it can make the animal lean and essentially get rid of its fatty liver disease, which is present in these mice, and also improves this glucose metabolism. Most interesting is this survival data here. So in 6 months, we started with about 20 mice, and in 6 months, we lost half the mice due to some of the other complication. But the mice treated with Glilo, not a single animal died. So we think there's something very interesting. This is an interesting approach that can be combined with other approaches that might be used for Alzheimer's to sort of improve the metabolic health of the animal. Next slide, please. So we're now actually looking at the effects of Glilo. What I showed you earlier was more a sort of a model of diabetes and obesity. Now we are trying to look at Glilo effects in normal mice. And we see, again, we're seeing some very promising effects. We do an assay for what's called to test motor coordination of the animal. These mice have been fed Glilo for like 6 months, and they're about 11 months old. So sort of, let's say in their mid-30s. And you can see the animals that are treated with Glilo have a 40% improvement in motor coordination. This motor coordination test is, I'd guess, equivalent to like an Olympian balancing on the balance beam. So the question you're asking is, how long can you really be on this beam? And we find that these mice can stay there about 40% longer. We've found that these treatment with this Glilo can also turn them into sort of endurance athletes. When you introduce a running wheel, you might have seen that hamster running wheel in the pet stores. We can also add that to the cage, and you can see that the mice treated with Glilo have more motivation to run and can run for much longer. They also improve glucose tolerance. So glucose tolerance here is measured by you give a bolus of glucose to the mice and you ask how quickly does it clear. And you can see here, these mice treated with Glilo are much more efficient at reducing the glucose, and it kind of goes back to normal within an hour. And finally, we're looking at now long-term survival. So we wanted to make sure that this is good for long-term health, not just reduce appetite and improve some of the health span characteristics. And the early data again is looking promising. This is Glilo given to a mouse, 2-year-old mouse, which is equivalent to a 60, 70-year-old person, let's say. And then we're finding like almost a 30% increase in the lifespan upon treatment. So, and full disclosure, now because these are commonly used supplements, these are now have been made available online on Amazon as well, if anybody's interested to find out more about it. Thank you.
Okay, thanks to you, Pankaj. Now I understand that we have made friends with Zoom, and now I'm going to introduce Dr. Julie Andersen. So Julie's a neuroscientist, that's how she trained. Her lab is focused on understanding the age-related underpinnings of both Alzheimer's and Parkinson's disease. So as I said earlier, before we lost Julie, here's what I think is really cool about her work. One of the projects is focused on boosting natural protective processes that tend to go haywire as we age. And one particular project involves looking at a metabolite that is derived from fruit as a possible treatment for Alzheimer's. So Julie, welcome, take it away.
Thank you, Kris, and my apologies for not the inability of the audience to see me, but if you would like to see what I look like, you can go onto the Buck website and go and look at up information on my laboratory. So next slide, please. As Kris said, our laboratory over the last several years has become very interested in what is called the gut-brain axis. So to a large degree, you sort of are what you eat, and what you ingest can have effects on the bacteria and other organisms that are in your gut. It's kind of amazing. The gut is made up of something like 10,000 different organisms that are involved in processing food and keeping the gut healthy. And it's known that during aging, people experience changes in their gut microbiome, and that may be due to genetics, aging itself, or really due to the types of foods you eat, et cetera. And that can impact on gut health in unhealthy ways, where you may experience an increase in gut inflammation. And there's been a lot of interest over the last decade or so, including the impact that gut inflammation can have on the brain. And it turns out there's sort of two hypotheses about how changes in the gut microbiome and gut health can impact on the brain. One is that when you have this change in gut bacteria and have more pro-inflammatory bacteria, you end up with what's called leaky gut and gut inflammation, and you produce pro-inflammatory toxins like cytokines that can pass into the bloodstream and into the brain and can promote neurodegeneration. Another means by which scientists now think that the gut can influence the brain. It turns out that the proteins which build up within neurons and near neurons in diseases like Alzheimer's disease, you've heard the term plaques and tangles. The proteins that make up those plaques and tangles, A beta and tau, which Tara has told you about their research in that area, are actually located also, they're expressed also in the gut. And when you experience a change or dysbiosis in the gut microbiome, that can elicit inflammation within the gut. And you actually can have a spread of those proteins up a nerve called the vagus nerve into the brainstem and other parts of the brain. So in something like 20% of a subclass of patients with these types of neurodegenerative disease, they end up having some inflammatory gut disorder, inflammatory IBD or something, maybe 10, 20 years before they start showing the loss in cognition or loss in motor function that are associated with Alzheimer's disease and Parkinson's disease, to the extent that that's looked at as a precursor before you actually see effects in the brain. So lots of interest in this gut-brain axis. Next slide, please. So what can we do about this? Well, it turns out that the body has evolved. It has a natural way of eliminating cellular junk. And that's not only junk associated with the buildup of proteins within these inclusions in the brain that drive neuronal loss and loss of cognition and motor function in AD and PD, but also you end up with an increase with aging and in these disorders with defective organelles within the cell. So what do I mean by organelles? Well, we kind of think of the nucleus as being the brain of the cell. There's another organelle, which you may have heard of, called the mitochondria. The mitochondria is the powerhouse of the cell. It's what produces all of the energy that the cell needs to perform all of its functions. And during the process of autophagy, these defective proteins and defective organelles are tagged, and they're taken to the cellular recycling center. And if you can transition to the next slide. This organelle is called the lysosome. And the lysosome is basically kind of an acid-filled sac. And when these defective cellular components of cellular junk are taken into the lysosome, they're broken down and they can be reused to make new proteins and new organelles. So next slide. So as a consequence of better understanding in the scientific community of the impact that change in gut bacteria and gut health with aging can have, we got interested in a particular compound called Urolytin A. So Urolytin A is actually what's produced from a precursor molecule that's found in a lot of health foods, including pomegranates and nuts and berries. So when people ingest these health foods, the bacteria within the gut converts the lactic acid found in these foods into Urolytin A. And then Urolytin A can enter the bloodstream, cross into the brain, and it causes an increase in autophagy or breakdown of these protein aggregates and defective organelles. So next slide. So I think we missed a transition, but that's fine, not a big deal. So we've been exploring, we've been exploring the use of Urolytin A in preclinical mouse models of Alzheimer's disease. I don't have pretty pictures like Pankaj does. We have them, but I'm not showing them here. We found that in mice, which we've been giving Urolytin A in the food, these mice normally develop plaques and tangles similar to what you see in humans, and also cognitive defects. And we can greatly reduce both the neuropathology, the Alzheimer's related neuropathology in the brains of those mice, as well as improve their cognition. So we're very excited about this. We want to understand better what the mechanism is that's driving these effects, because this could lead to the development of novel therapeutics. I should point out that Urolytin A has been tested in just normal aging mice. And actually there's a small clinical trial that was published in 2017 in humans. In the small human clinical trial, when Urolytin was given to sedentary but otherwise healthy elderly individuals, it actually, over the course of even a few weeks of treatment, improved muscle function in those individuals, so higher running speed, et cetera. So it seems to, the good news about this is that it seems to be safe, at least over this smaller time period in humans. I think larger clinical trials are underway looking at more chronic effects of treatment with Urolytin A. And also it may have like multiple effects in terms of healthy aging. And we're really interested in exploring its effects in terms of keeping the brain healthy during aging. So thank you very much. And again, apologies that you're not seeing my picture, but such are the vagaries of Zoom, even after all this time. So thank you.
Thank you, Julie. So yeah, Zoom, yikes. So I'm going to get to questions now. I'm seeing them coming in. I'm going to review them. Before we get to the questions, I have a question. I would like in particular Julie and Tara to just talk briefly about their take on this new FDA approved drug for Alzheimer's. I think it's called Helm. It is really controversial. What do you think folks need to know about this drug?
So I think, so I'll take the first shot at this, Kris. Yeah, for people who aren't aware, there was Helm recently received expedited FDA approval as therapeutic for early stage Alzheimer's disease. And it's really an immunotherapy to get rid of A beta within the brains of early stage Alzheimer's disease patients. There have, since that expedited approval, there have been concerns raised. There's been pragmatic concerns raised about how the drug should be administered. And there's been concerns about the fact that it's not clear that insurance will pay for it. And it turns out to be quite expensive, something I think like $6,000 a month. And to be honest, a lot of physicians and scientists have raised concerns in the sense that it's not clear that real efficacy was shown in the studies that were considered as part of the FDA approval. And in fact, the advisory board that looked at all of that data voted no on FDA approval. So it has raised a lot of concerns within the scientific community and amongst physicians that may consider prescribing this. I'll let Tara have a crack at answering.
Great. Yeah, thank you, Julie. Yeah, I would just add that I mean, Helm, it seems like is effective at reducing amyloid beta people in the brain. So that's good. However, the most recent phase 3 clinical trial that was done was interrupted in the middle of the trial because it looked like the results were suggesting that it wasn't necessarily having a beneficial effect on cognition, at least in some of the patients that were taking it. So the trial ended early, and that's maybe not typical for a FDA approved drug. And so that's why the FDA gave the, the accelerated approval. However, there is a requirement that the company needs to perform another clinical trial to address whether Helm actually helps people or has a beneficial effect on their cognition. And I think that would be really important because just lowering amyloid beta levels doesn't necessarily mean that it has a beneficial effect for patients' daily living. So that will be important to find out.
And just to kind of add to what Tara's said, this is Julie again, and we may, Kris, get to this, but in terms of Alzheimer's disease, we still are really trying to figure out it's very complex disorder, and we're still trying to figure out what all the components are, if certain components are involved in causing other components. Certainly, A beta aggregation is something that seems to occur early on, but how much it's involved in later pathologies, but like tau or neuroinflammation, and to what degree, we don't really know. So, and it's not clear at all that this would be a good treatment for people with moderate or severe Alzheimer's disease.
So based on what you just said, Julie, I'm going to go to a question that we got from somebody because it's sort of related. So there have been questions about whether both A beta and tau, whether they are causal for the disease or whether they're symptoms of the disease. Where's the science with that right now? What can we say? And we have lots of questions, so try to keep your answers brief because I want to get to as many of them as possible.
So I think I can just mention something. We know that there are mutations in the APP gene that cause Alzheimer's disease. So this APP gene is where amyloid beta comes from. So we know that a single mutation in this gene can cause Alzheimer's disease. So amyloid beta is sufficient to do that. So it does cause Alzheimer's disease. In the case of tau, a single mutation in the tau gene is known, can be known to cause familial dementia. So tau can cause dementia. We have evidence of that in humans. We've also do mouse research to study these genes, the tau gene, the APP gene that causes dementia.
Yeah, and again, this is Julie. I think carrying on from Tara's point, there are these rare familial mutations which are associated with early onset Alzheimer's disease, like the mutations that increase A beta levels. But I think it gets a little bit murkier when you start looking at late onset disease where maybe it's not a particular familial mutation, but it's probably a combination between genetic susceptibility, aging, and environment. I think it's a little harder to predict what's going to happen, and those cases of late onset constitute something like 95% of people with Alzheimer's disease. So I think it's the studies on the rare mutations that's really important because they likely do cause disease by pathways that also play a role in sporadic. It's just more complicated to really understand in sporadic cases.
Got it. So PK, here's one for you. Somebody wants to know about Metformin and whether that might be helpful as a preventive measure for Alzheimer's, like what kind of protection it might have. I'm going to talk, I want you to talk about Metformin, what it is and what it does in the body, and then maybe Tara or Julie can jump in about the brain.
So Metformin is probably the most commonly prescribed drug for type 2 diabetes now, and has been shown to have shown to not only keep glucose in control, but also potentially improve health span. And I want to say it's a great question, and it's as you'd expect, people have done clinical trials on this. And one of the things that I saw recently, which totally surprised me, was they found that so you can imagine an anti-diabetic drug could act differently in different parts of the body. So the data suggested that there was improvement in metabolic health and glucose, but it did not reverse some of the symptoms in diabetes on the brain. And one of the more related to memory and dementia. So I want to add that I think we're still trying to understand how metabolic disturbances influence different parts of the body, and where some treatments could be relevant for reversing most of them, but also some may only be relevant for parts of or may have segmental properties. So I was actually I thought it would work, but I was surprised that it didn't work as well. I'll let Julie and Tara answer more on that.
Yeah, in terms of metformin's effects on the brain, I know that there are some studies that suggest that it promotes stem cell proliferation or differentiation. I'm not really sure what, but I to be honest, I'm not familiar of clinical studies that have been done that specifically. They probably have been looking at cognition. Tara, do you know, or PK?
I think there have been some small, maybe relatively small studies done, and potentially with some mixed results as to how beneficial. But yeah, that's the extent that I know.
Yeah, and I do know that there are some animal studies that came out a couple of years ago that suggest there may be gender specific effects, like working in women and not men. And that's also something, fortunately, that the NIH is paying more attention to. In the past studies, there tended to be more men involved in clinical studies than women, and they're trying to balance that more and to understand more about gender differences. And that actually includes in preclinical studies, like the kind of studies that we're all doing in mouse models, looking both in male and female to see if there are differences in terms of different types of intervention or drug treatments.
So we got a question about brain inflammation and what brain inflammation, what impact does it exist, and what does that do for possible Alzheimer's? I know that's a focus in your lab as well. Julie, do you want to kick off the conversation about brain inflammation?
Yeah, this makes me think about the previous question from the audience about whether A beta and tau are just simply hallmarks of the disease or causative. I think Tara's response in terms of the familial forms where you are expressing higher levels of A beta do suggest that there's an involvement of those components. But I think in the last decade or so, there's been a growing realization that increases in neuroinflammation in the brain plays an important role, if not in initiating the neuronal cell loss, certainly in propagating, and particularly activation of a class of inflammatory cells found in the brain that are called the microglia. So there's been a lot of studies towards looking at the effects of microglial activation, inflammation produced by that. In terms of Alzheimer's disease, I think there's some good data coming out that backs that up.
Anything to add, Tara?
Oh, thank you. Yeah, I actually just wanted to, I saw a question in the question and answer, and I do want to make sure people understand this genetic concern that, as Julie said, 95% of people who have Alzheimer's disease, we do not know the real cause of it. We don't have a real genetic cause. Of only a very small number of people have these familial mutations that cause Alzheimer's disease. So the vast majority of people, we don't know. There are genetic risk factors, such as ApoE4, but if you have ApoE4, that does not necessarily mean that you will get Alzheimer's disease. It's just a risk. It increases the risk. So I hope that clarifies that.
Yeah, and I would say that actually makes environment all the more important, right?
Sure.
It's like I sort of, in my family, we are genetically predisposed for cardiovascular disease. I had a grandfather who passed away in his 50s. My father had high cholesterol. However, and I've inherited that risk, and so I need to be careful about what I eat, to exercise, etc. I think knowing what your familial predispositions are is useful in that regard. Yeah.
So I just want to be clear here. So because I have some Alzheimer's in my family as well. I don't think we have the ApoE4 gene, which is the super risk factor. But if one has a familial tendency towards, let's say, diabetes or high blood pressure, if there are trends in the family, that doesn't mean that you're going to get Alzheimer's. Or if somebody else in the family has late onset Alzheimer's, that doesn't mean you're going to get Alzheimer's, but there could be some risk factors that you need to pay attention to. Is that the best way to understand it?
Right.
Yes.
And I think PK could, because part of what Pankaj is addressing is this link between, as you were saying, Kris, diabetes and Alzheimer's disease. So I don't know, Pankaj, if you want to elaborate.
Yeah, plus Pankaj, there's a question. Somebody wants to know the ideal diet to prevent Alzheimer's. So you can kind of do a combo there.
Yeah, I think I would say, like you said, I'm not a clinician, so I hate to give advice on that. I'll just tell you what I personally do. I think from the different studies that have come around. I think, for example, one thing that shocks me, and I would ask the people to ask themselves, how much sugar do you think we eat compared to what we'd had 100 years ago, right? So if your answer was one, twofold, fivefold, you're wrong. We're almost eating 50 to 100, 10 to 50 times as much sugar per capita than we were 100, 200 years ago. So that just tells you how we have altered our diets and how things have shifted a lot from what they were before, and we're still trying to understand what's the right diet for us. A lot more research is needed for this, but more and more data points to having a diet with fiber, increased fiber, and and reduction of any added sugar. You don't need any added sugar in your diet. That's just something we have created. So whatever you can get from foods, that's how we should be eating. Right? So and then complex carbohydrates are not. Are a good thing as well, because they release the sugar if whatever there is in a slow manner. And I've seen some studies that even just adding a few portions of fruits and vegetables to your diet has very strong protective effects even in Alzheimer's. So I would, so that's my recommendation. More keep, if you're taking sugar, it should be definitely moderate, but wherever you can avoid it, yes, that would be the way to go. And yeah, so is anything else I left, or?
I think you got it. I'm looking at the questions too. So Tara, somebody's really interested in your qibra protein. Is there anything that we can eat? Is there anything that we can do to boost that guy? What do we know about, are there any actions currently associated with your research?
Thank you. Thank you for asking that. Not that I know of. Not that I know of yet. The way that we've currently been studying how we can introduce qibra to the brain would require gene therapy. We are also considering maybe peptide therapeutics to look into that. But in terms of using nutrition to boost qibra levels. That could be definitely worth looking into in future, but not that I know of currently.
Okay, we've gotten some questions about the use of statins. So statins have been, I know, implicated, or there's been talk about the effect of statins on the brain and on brain health. Anybody want to tackle this? What should people know about statins and Alzheimer's disease?
I know that there are some preclinical studies that have suggested that they're beneficial, and I assume clinical studies as well.
Tara, anything to say about
Yeah.
statins?
I'm not as familiar with it, so I'm not sure I can comment.
No problem. So there are some questions about a very popular supplement that is being used to address aging. There's many supplements, but it's based on this molecule called NAD+. And people are wondering, and then it's also related to resveratrol, which is a popular supplement out there. So I'm wondering if there's anything to say about that. And I guess the larger issue is there are a lot of supplements out there that are available to people to take that may or may not work. How do you have any of you, do you have take on that? How should people approach this topic?
Yeah, I think it's always important. So a lot of supplements are, our supplements are not regulated by the FDA, and they could potentially interact with prescription drugs, So I think it's always important before you start taking any supplement to discuss it with your physician, even just so that they're aware what else you're taking in addition to your prescription medications, because you don't know, and I think it's important to discuss this with your doctor.
Yeah, I agree with Julie. That's the first thing I would do personally.
I mean, unfortunately, there's a lot of things where there's some levels of some things may be good, but only up until a certain level. And sometimes you can get too much of a good thing. This is preclinical, but we have some data in terms of looking at high dose vitamin D in mice, in Alzheimer's mice, that they're more likely at this very high dose to become pre-diabetic. So what that means to humans, we don't know. The mice are not humans, but it's sort of, I don't know, personally given me pause about taking any type of really large dose of a supplement. Definitely check with your physician.
So let me take this maybe to a broader thing that all of you can talk about. So a lot of the supplements out there, like, we got a question about NAD+, are really directed at the aging process and slowing the aging process, so we don't age as fast. So the buck is very, very focused on the aging process. And, our hope is that if we can really slow and impact the aging process, we can address a lot of different age-related diseases in one fell swoop. But talk about when each of you, when you think about the aging process, how does that impact your research on Alzheimer's or how you look at Alzheimer's? There are researchers around the world who are studying Alzheimer's. Does the fact that you and we have a focus on aging, does that impact how you look at this disease?
Yeah, I want to start by answering that, because for me, I just want to say that we were studying diet restriction and aging for almost 20 years. And as there's the interest in AD increased. And I want to show how this is an example of how, when there's funding provided, how it changes research. As funding came through the NIA for this question, literally every project in our lab we haven't changed our direction. We're still studying the same molecules and pathways, but we just started asking whether it would be also relevant for AD. And to our surprise, and I have to say, it's really motivated my lab a lot, because we're finding, oh, this thing that we were studying has relevant to a disease. And so that really motivates us a lot. And has really pushed experiments. But this is a very good example where, having the research funds to do these experiments forces us to look under the carpet and say, oh, well, maybe it's also linked. There might also be a link here, and we've been very successful with this approach. One of the examples I just want to give is, like, we've found again another example of glucose metabolism, which we're collaborating on with Tara and Julie, that there's a little battery in your for glucose. There's a battery which is essentially the glycogen, right? So glycogen is accumulation of glucose, and when glucose runs out, you utilize glycogen. We're finding that in Alzheimer's, even in fly models, there's defects in using this, being able to access this glycogen. So we're very interested. I mean, this was more a question from an aging aspect. Now it's turning into a question that's relevant for dementias as well.
Yeah, I think that definitely in the last, so my training is as a neuroscience scientist, although my first faculty position was in the College of Gerontology at USC, and then I've been at the Buck for 20 years. And I think that has really molded the way that I think about Alzheimer's and Parkinson's. I think historically people have tended to study disease in silos and thinking about the disease itself, and particularly for age-related disease, not really paying attention to what it is that kind of happens globally with aging that could influence particular disease processes. And I have to say that the idea that aging is not only a risk factor for disease, but is likely the processes involved in aging are underlying drivers of almost all age-related disease. There was a term coined in 2007 called Geroscience, and I'm proud to say it was coined at the Buck Institute as part of a large center grant. And this is really, over the course of the last 15 years, been picked up worldwide, including by the NIH. And there have been a lot of symposia around this. And I think that this has really revolutionized the study of age-related diseases, including Alzheimer's disease, and looking for novel therapeutics.
Yeah, and I want to give full credit to Julie. I remember when I was a graduate student, there's very few neuroscientists who show up to aging meetings and vice versa. And Julie was one of the first few, one of the few who I've interacted with for the last 15, 20 years.
You make me feel so old. Yes, I was fresh new assistant professor when I met Pankaj, who was a graduate student in Manchester.
Yeah, and I want to say now that we're seeing a lot more interaction between people who study aging and neurodegeneration, and I think what I am excited about is that all these approaches are going to complement each other. I think the neuroscientists, there's no wrong answer here. There's just going to be more approaches focused at this question and people looking at it from different angles. And I think people are going to find more information is needed, and I think it'll translate into better therapeutics for people.
That's great. So here's a question that I think talks about a lot about in aging, and all of you might have something to add. So I got a question about exercise, and how does exercise impact brain health? How does exercise, impact Alzheimer's? I know in your lab, Pankaj, I see those fruit flies jumping up and down inside the vial when they haven't had enough to eat. So what can you tell us about exercise and brain health? Julie, Tracy, what do you have to add?
Yeah, I think there's a lot of anecdotal evidence towards exercise. I'm not aware of any kind of large, in the context of Alzheimer's disease, I'm not aware of any large clinical studies that have been performed. There's probably better clinical evidence in terms of Parkinson's disease and exercise. But you know what? Exercise is good for you, and it's good for the whole body. And I don't know, anything that's good for the whole body, I suspect is a good deterrent for aging, including brain aging.
Yeah, I think I want to just add one aspect to this. Our lab is now studying a lot about genetic variation, and you saw Tara's presentation as well. There are a lot of these interventions, I'd say fasting and exercise, and also the different drugs that are out there. There's going to be a very important aspect of individual variation. So, I know personally, the days I exercise, I'm a different person. It's like, it's day and night for me. And it's always been like that. So I think some people respond more or less to these things, but generally I would say they are going to be protective, and it's almost not a question of if, I think almost a question of how much they're going to benefit, and how, are you at the ceiling, or is there more room for you to optimize this? But I think the more we can do, the more the natural interventions, the better I think we are, the better we will be sort of at avoiding the long-term sort of consequences or decline. Yeah.
I don't know, Tara, what do you think?
Yeah, I agree. I think there's maybe been some small studies on humans, some that show beneficial effect and some that don't. So again, that's kind of frustrating, like knowing whether this could really help people. And as exactly what Pankaj said, it might help some people, it might not help other people, depending on other risk factors. So maybe that's something, this is something to take in consideration in big clinical trials. And I just want to make a plug. We can figure out these risk factors if we have better biomarkers to determine how likely people are to be susceptible to Alzheimer's disease. So that's something else that's, I think, really important for
absolutely. And that is going to help a lot with personalized medicine too, if we had biomarkers. Yeah, for sure.
So we are nearing the end of our hour. I just want to ask each of you just to take a look ahead, like let's say 20 years from now, and look at where would you put your bet where we're going to be with Alzheimer's? Is there ever going to be a silver bullet for this disease? Are we going to be able to, quote, get rid of it? Or will we just, like, for those who have it, or will we just slowly be able to prevent it? What's your, briefly, what's your outlook on what this is going to look like, let's say 15, 20 years from now?
It's hard to predict, but I given that people are recognizing more and more that, exactly what Tara was talking about, that there are, and Pankaj, that there are different, for each individual, there may be different genetic risk factors or different environmental exposures, appreciating that there may not be a silver bullet, and but there may be things that are going to be useful on an individual basis, and it may be a combination of silver bullets for each person. So I think the more research that we have, and the better understanding that we have of the very heterogeneous nature of the disease and how individuals may be impacted, I think the more of a shot we have of coming up with those, I don't know, silver bullets.
Tara, what do you think when you look ahead?
Thanks. Yeah, so I hope we have different approaches to treatments and therapies. In the past 2 decades, there's been mostly focus on amyloid beta targeted therapies in drug development. And we all know that that hasn't been as successful as we have hoped. So we need to target other potential risk factors or things that could drive the disease. I know there's been some development of an approach to target tau. So I'm excited to see what those results are, to see if reducing tau levels in the brain can ameliorate cognitive impairment. And there are, of course, other things in the pipeline that we're all working on that, I'm excited to see if kind of a multimodal approach to treatment will benefit people based on their individual risk factors. So I really hope that's where we're heading rather than having a silver bullet.
Pankaj, you get the final word here. So what do you think it's going to look like?
I am naive and optimistic. I think we're going to have a lot better understanding of prevention and treatments. But though I have to say, one thing that sort of makes me concerned, I think we'll have all that. And just like we are seeing in obesity and diabetes, the incidence is going to keep going up. So even though there are good ways to sort of overcome this and there will be better treatments, the question is, will they really reach the average person and everyone? That I'm not as optimistic of the human species, but I am optimistic that our understanding will improve, and I think we should be able to prevent or delay it in cases in people who are motivated to do so.
Great. All right, that does it. Thank you very much to Julie, Tara, and Pankaj for taking time to be with us today. Thanks to all of you who attended and participated. We're going to save the Q&As that we got, and we will attempt to answer as many of them as possible when we send out an email once this video is up and ready for posting. So just keep your eye out for that. And before we close, I just want to let you know that I would be remiss if I failed to do a fundraising pitch. Here's my message. Your donations matter. All of the projects discussed today started with the seed of an idea. Each of these scientists have benefited from and are grateful to donors who have provided the support that allowed those ideas to grow. There are more seeds waiting to sprout. I work with these folks, they deserve your support. So I encourage you, before you get on with the rest of your day, to go to our website and enjoy being generous. You could specify donations for Alzheimer's research. You can choose to support any of these researchers individually, or you can simply support the Buck's overall effort to help all of us live better longer. So thanks for being with us. A short survey is going to be popping up on your screen as we leave the event today. Please take a few minutes to share your thoughts. Thank you.